So welcome to the IB seminar here at OSU. I'm very pleased to introduce to you Dr. Tyrone Hayes, who will be speaking today. Dr. Hayes is a professor at the Integrative Biology Department at UC Berkeley. He earned his bachelor's degree from Harvard University, received his PhD in biology from UC Berkeley in 1993, and then held an appointment there, has held one there since finishing his doctoral research. Tyrone's work centers on the role of endogenous steroid hormones in amphibian development. He also examines how amphibian development is affected by endocrine disrupting chemicals, such as the herbicide atrazine. Basically, he's shown that even at low concentrations, atrazine turns male frogs into female frogs, leading to the degradation of amphibian populations and a decline in environmental health. Dr. Hayes has also done important advocacy work in environmental racism. He's shown that endocrine disrupting pollutants like atrazine can affect public health, especially the health of ethnic minorities and people of lower socioeconomic status who are at greater risk of exposure. And he's also involved in advocacy work that highlights systemic racism in STEM. He was one of the leading signatories on a letter to Science magazine that just came out this September, calling for an end to systemic racism in higher education. And with that, I will turn it over to Dr. Hayes. Make sure you use the Q&A board to post any questions you may have, and Dr. Hayes will periodically pause to address those questions. Dr. Hayes, we're very pleased that you could come talk to us today.
Thank you, thank you. It's my pleasure. So if I can share screens. I think that went smoothly. Oh, I think nothing's happening yet. Okay, there we go. It's my pleasure to join you today, and I want to thank everybody for coming. Although I've certainly been giving lectures and seminars quite a significant part of my life, I'm new to Zoom. This is maybe only certainly less than a dozen Zoom talks, and for some reason things tend to go longer on Zoom. So I'm flexible. We can pause for questions as we go, and I've loaded slides that we don't have to get all the way through. We can stop and have discussion anytime you want. It's your time. I was told that there was an interest in sort of addressing science and society. So rather than a typical talk where I'll go through the latest, hottest thing going on in the lab right now, this is going to be kind of like a wedding, something old, something new, something borrowed, and we'll have to look for something blue, as the saying goes. Before I start, I want to start with my acknowledgements. So I never present myself or my work without acknowledging the people who made me who I am. And this is an old picture, but first and foremost, I owe a great deal to my family for their love and support, my parents and my brothers and my wife and my kids, all of them. And I have to thank the funding. So I'm going to talk about work that's been ongoing for a little over 20 years. And we've had a lot of different funders. And this is also my disclosure. I have been funded by the chemical manufacturer Novartis, but a couple of decades ago, they decided not anymore. There's an old saying that goes, you know you're doing something important. You're not doing, if you're not pissing somebody off, maybe you're not doing anything important. By that measure, I'm pretty important, because I had some people pretty pissed off at me. People who I hope are not pissed off at me are my students. So I want to thank all of the students that have been involved in the work. And I want to point out that everybody in blue, and these are various iterations, everybody in blue is an undergraduate. And this is the most recent picture I have, pre-COVID picture of my lab. And one of the other things that's been really wonderful for me is that I've had the opportunity to work with an amazing diversity of students, and diversity of all kinds, socioeconomic, ethnicity, background. But probably most important here, I've worked with a number of students whose parents work in the agricultural industry. So it gives me a whole new perspective to be studying a chemical and how it impacts frogs, and working with students who are saying, wow, my parents were sprayed with this chemical most of their life. And finally, I want to dedicate this to my grandmother, who passed away in 2005. But before she did, she passed on to me her love of education and her desire to make the world a better place through education. She also taught me something that you can't learn in fancy places like Harvard and Berkeley. And that is, if you want to get across, if you want to get a point across, don't preach, don't teach, just tell a good story. And I like to think that that's my job here, is to tell you a story about a little boy who likes frogs. First and foremost, that's who you're speaking with today. And that would be the beginning of my story and the end of my story. I've been showing this slide for 28 years now. My mom mailed it to me from South Carolina, where I grew up, when my son was born. And she had written a note that said, "This was your dad's favorite book." She gifted it to my son. I don't remember the book, but I've been in love with frogs, and I've been trying to answer this question for as long as I can remember. Now, I tell you the whole story, but that involves 53 years, and I only have about 50 minutes. So we're going to skip a few years through my love of frogs and my life as a little boy who likes frogs, and we're going to go straight into graduate school. Because graduate school is where a long-term childhood dream of mine came true. As a kid, I dreamed of going to Africa. I remember my dad bringing home National Geographic magazines, and I remember folding out the maps and dreaming of going to this magical place. And truly a dream. I never got on a plane actually till I went to college. So, and the likelihood of me going there prior to that was almost zero. I got to go in graduate school, and not only did I get to go to Africa, National Geographic paid for it. I became a National Geographic Young Explorer. I got to be on the magazine. I got to be on the show. Literally, if you can imagine a dream come true, if you can imagine that thing that you dreamed about when you were a little kid, this is what that experience was like to me. It had a big impact on my life. I actually went there to study African bullfrogs. That project didn't work out because I couldn't culture the frogs in laboratory for a variety of different reasons. But while collecting frogs there in the Arabuko, I believe you can see my arrow there. It's along the coast in Kenya, along the East Coast, along the Indian Ocean. And there, even though I was not successful with the species that I went there to study, I was introduced to Hyperoleus argus, this so-called reed frog, where the males and females look like they don't belong to each other. They look like completely different species. So I became interested in why they were differently colored. My undergraduate training was with a man named Bruce Waldman, and he was a behavioral ecologist and an evolutionary biologist. And so I became interested in things like, what are the males doing in their environment that's different than the females, that they would have these different colors. And there was some suggestion that the female bright coloration was aposematic and that they potentially were poisonous. I don't know about all that. But in my graduate work, I was a developmental endocrinologist, and I mostly worked and tested hypotheses in the laboratory. And so with that training, I decided to try to understand not just why they were differently colored, but how they become differently colored. And these are the kinds of questions that are on a little boy who likes frogs' minds. So first off, we brought the frogs back to the laboratory. And this is the same frog photographed once a day for 6 days. And so our first really simple discovery was that they all start out green. Like literally nobody had raised these animals and knew that at the time. And at puberty, at sexual maturity, the females change color. In the laboratory, that's in about 6 or 7 months, the females will start to change color. So we hypothesized that color change was estrogen dependent. So in the same way that in humans, when you reach puberty, if you are an XX individual with ovaries, you express estrogen and your breasts grow. And if you're an XY individual with testes, so in that same way, we predicted that estrogen would induce this color change. And so we tested that the way any little boy who likes frogs would. We literally just dipped the frogs in hormone solution. And if you dip them in testosterone, nothing happens. So testosterone is the so-called male hormone. But if you dip them in estrogen, and this is the so-called female hormone that's in every vertebrate animal. So it doesn't matter if you're a fish, a frog, a dog, a cat, a hog, or a human. If you're a reproductively functioning female, this hormone circulates in your body. Made the exact same way, works the exact same way in all vertebrates. And it made our frog change color. So if we dip them in estrogen, you can see this is, again, a frog that's been dipped in estrogen. You can see they'll start to change color. Now, this had a profound, this is rather simple experiment, had a profound impact, as did Hyperoleus argus, on my career and my trajectory in ways that you couldn't predict. So we ended up, believe it or not, We ended up patenting this frog. It was my wife's idea. We call it the Hyperoles argus endocrine screen, or the Hayes test. And we patented the frog. First and foremost, if you're a little boy who likes frogs, you patent a frog because it's cool. Then you say, hey, I have a frog. It's my frog. Here are the reasons that scientific reasons that you patent the frog. So here's the control, an untreated animal. They all start out green, okay, at metamorphosis. If you're exposed to the estradiol, the natural estrogen that's in all vertebrate animals, from fish to humans, you name it, they'll change color. We screen dozens of other compounds, such as ethinyl estradiol. This is a synthetic estrogen that's used in birth control pill. They'll change color. If you expose them to DES, another pharmaceutical estrogen, they'll change color. If you expose them to DDT, an insecticide that happens to mimic estrogen, a so-called endocrine disruptor, they'll change color. And so the bottom line is we screened dozens of compounds, and we found that every compound that made my frog change color was also known to promote breast cancer. And so with breast cancer being the number one cancer in women, and with cancer being the number one cause of death behind heart disease, we potentially had a very important little frog from the Arabuko where we could screen chemicals, or we could screen water. You could send me a sample of your water, could dip my frog in it. If it changed colors, well, then you might want to reconsider your water source. The other thing we showed was that we could pre-expose these frogs to tamoxifen and block the effect of estrogen. And tamoxifen at the time was the estrogen blocker that was used to treat breast cancer. So we had a frog where not only could we detect potential breast cancer causing compounds in the water, but we could also pre-screen and test compounds that might be used in the treatment of breast cancer. So a little frog could potentially give us both kinds of answers. To give you an idea of, I think I entitled When Frogs Become Canaries, with the idea being that the frogs are telling us something about humans, which is not why I studied the frogs. I studied the frogs because I'm a little boy who likes frogs. But to give you an idea of how relevant it is, estrogen requires the estrogen receptor in order for you to get a response. In humans, as in other animals, androgens like androstenedione can be converted into compounds like estrone, which can be converted to compounds like estradiol. And once it binds the receptor, they can do things like make your breast grow, or make you have your menstrual period, or as you get older, induce breast cancer or promote breast cancer. What's more is estrogens are metabolized in some interesting ways. There's so-called catechol estrogens, some of which, such as 4-hydroxyestradiol, can still bind the receptor in humans. Then there are compounds like estradiol 3-sulfate estradiol, which can be converted back into estradiol. But then there are compounds like 17-sulfate estradiol, which can't be converted back into active estrogens. And so if you look at the human condition, there's several points shown by the red arrows that if we could block those pathways, we could find new potential treatments for breast cancer in humans. And so it turns out our frog maps exactly to where you'd want issues in frogs. So they don't respond to androstenedione. Androstenedione also does not promote breast cancer. They do respond to estrone E1. They respond to E2. They respond to estrogen unless you block the receptor, as we did in this case. They respond to the 2-hydroxyestradiol and to the 4, okay, as you might predict. But down here, they don't respond to the 17-sulfate estradiol, and breast cancer cells also don't respond, but they do respond to the 3 version of the estradiol. So we had a frog that essentially shows the same specificity to estrogens as human breast cancer cells. Now, maybe now's a good time. Maybe now I can pause if there's specific questions, because I'm not going to talk more about that part of my lab. We went on to do some evolution-related questions, but that's all I'm going to say about hyperolia. So if there may be questions now, I don't know how I would find them. So if somebody wouldn't mind finding reading questions, if there are questions, and if not, I just give me the signal and I'll move on.
So Tyrone, there are no questions so far.
Okay. So here's what remember, I'm a little boy who likes frogs. And for me, it was just neat, to be in Africa, my dream come true, and dipping frogs in solutions and testing hypotheses at the same time. The university had a slightly different idea. I like to say I was introduced to a grown-up word. in this case, the grown-up word was intellectual property. So once we filed the patent, the intellectual property turns out, I don't know if I'm assuming all faculty know this, students might know this, might not know this, but your intellectual property actually belongs to the university. And their stance was either show us how you're going to make money on it, or we're going to sell it to somebody else. And so my wife, who had an MBA and an MPH at the time, and we actually started a company using the lab at UC Berkeley, and we got our first customer, the largest chemical company in the world, Novartis, said, well, we want to know something about atrazine. I never heard of atrazine. Here it is. So this was a little over 20 years ago. So now I'm a new assistant professor now, and these guys were paying me more money than I'd ever seen in my life. And so they wanted me to test this compound that's an herbicide, atrazine, used since 1958. We use 80 million pounds. At the time, it was the #1 selling agrochemical in the world. It's used in more than 80 countries, so it's kind of a global issue, but it's now outlawed in all of Europe, which is interesting because the company that sells the stuff is based in Europe. So we're using 80 million pounds of something that's not allowed on the continent where the company calls home. So we got asked to use another African frog, the African clawed frog, Xenopus laevis. I never thought I would study Xenopus. It's the most common frog used in biology and developmental biology. For an interesting reason, in 1920, somebody discovered that the human pregnancy hormone, HCG, will make this frog lay eggs. So by 1940, this species was literally the human pregnancy test. If you thought you were pregnant, you would go to the doctor, they would inject urine into the frog, and if it laid eggs, you were either happy or sad, depending on your situation. So again, this is another idea of when frogs become canaries. This is another idea where frogs give us a warning or early detection about our own physiology, and that's why I tell the story. I tell the story also because it shows you, well, I mean, I guess there's 3 reasons I tell the story. One is it shows you the value of curiosity-based research. I still have not tracked down the story behind the first guy who thought, I wonder what'll happen if I inject pee into a frog. I have no idea why anybody thought to do that in the first place, but it was a very important discovery. The other reasons that I tell you this idea of when frogs become canaries and the early warning is it also shows you the similarity between frog hormones and our hormones. So my color-changing frog responds to the same estrogens that promote breast cancer in humans, and this Xenopus African clawed frog, it's our pregnancy hormone, human pregnancy hormone, is so similar to this frog's that it'll make this frog lay eggs. The other funny story I like to tell, or I guess it's not funny if you're an ecologist, but people threw these frogs out when they developed new pregnancy tests. So in fact, now all of my African clawed frogs that I study come from San Diego. So I call them my African American clawed frogs. I probably have the only African American clawed frog colony in the US. So at any rate, we use these African clawed frogs, and we showed while under contract with the company that atrazine inhibited growth of the voice box. And for those of you who don't know, male frogs sing or give advertisement calls, and female frogs don't, for the same reason that men in general have deeper voices than women, testosterone. So these data that atrazine inhibited growth of the voice box, and this was about 20 plus years ago. It was before I was still an assistant professor. So once we discovered that, we went to the gonads, because if you have low testosterone, then there's probably something going on with the gonads, the source. And so here's an animal, here's kidneys, for example. And here's an animal that has testes, that has ovaries, that it has more testes, that it has more ovaries. And this is not normal. And I don't mean that in any derogatory way or in any kind of judgmental way, but frogs are not naturally hermaphroditic. Despite what people may have learned, people always ask me, aren't frogs naturally hermaphrodites? Because apparently this was in Jurassic Park, that frogs naturally change sex. There is no real scientific data that I know of that frogs change sex or that they're hermaphroditic. There are fish that do that, but not frogs. This was not a normal aspect of development. The company also spent some time trying to convince the public that this was a naturally occurring phenomenon and I hadn't discovered anything bad about atrazine. And that's not the case. So we hypothesized the following: testosterone, the so-called male hormone, should come from the testes. In fact, the word testosterone is a portmanteau. It means testicular hormone. So it's the male hormone. But it can be converted into estradiol, into estrogen. So our hypothesis was that atrazine turns on the enzyme, the machinery that converts testosterone into estrogen. As a result, you become demasculinized because you're using up your testosterone, and you become subsequently feminized because now you're making the female hormone when, if you're a genetic male, you shouldn't be, at least not during development. And so we measured testosterone levels just like you can measure them in humans, and we showed that atrazine treated males have reduced testosterone so that they're not different from females. And at that stage, we published a paper. We called it Hermaphroditic Demasculized Frogs After Exposure to the Herbicide Atrazine at Low Ecologically Relevant Doses. And we published it in Proceedings to the National Academy of Sciences and PNAS, which was a big deal to me. It's such a big deal that I called home and I told my mom, I was coming up for tenure, and I said, Mom, I have this paper coming out in PNAS. My mom said, you publish papers all the time. I said, yeah, but this paper's coming out in PNAS. And my mom, in addition to saying, how did you get a paper cut on your penis? Because PNAS meant nothing to her. And in addition, a week later, my mom calls me back and said, how important is that paper? I said, what do you mean, Mom? She said, I'm with the Barnes and Noble. They never heard of that magazine. And it gave me a whole new perspective on what's really important, that here in the ivory tower, we reward each other and promote each other for things that mean nothing to 99.9% of the world. And so I've reshaped now what's important to me. And in fact, I will show you a kid's book, another publication that actually means a lot more to me now because my mom can buy it in Barnes and Noble and tell everybody what I'm doing. So, and by the way, 4 Black men and a Latina co-authored that paper. It's probably a record for the Proceedings in the National Academy of Sciences. As important as it was for all those reasons, and I did get tenure, by the way, there were some questions that weren't answered. Like, we didn't know if the hermaphrodites were males with ovaries or females with testes, because frogs don't have sex chromosomes. And at the time, we couldn't tell who was genetically male and who was genetically female. We also didn't know what happens when the animals become adults. So what happens when they grow up? But at the time, it took us about 5 years to get these frogs to adulthood. And, it's difficult to get an undergraduate, which is mostly what I work with, or even a graduate student, say, hey, I have a project for you. And maybe by the time you graduate, we might have interesting data. In fact, it was longer than that. It took like 8 years, but I had no tenure. I mean, I had tenure, so I had time to figure it out. By the time the paper came out, and it was another PNAS paper, a gene had been identified by a lab in Japan that we collaborated with that was only present in genetic females. And here's a lovely couple. And so we were able to use, only females have this gene shown up here. So sometimes we do fancy stuff like PCR and stuff. So we were able to show that, of course, this guy's a male, the one who looks like he's smiling there. And that's also a genetic male. That's his brother. And in fact, these eggs, actually, this is a really neat story. These eggs right here, 4 generations down, these eggs have now produced 4 generations. And we're submitting another paper in the next month or so from PNAS on what happens 4 generations down from here. So we kept these eggs and kept the experiment going for another, I guess it's been another 10 years. And we can talk about that in the end if you want. So at any rate, I could have published that. Genetically male frogs turn into females and laying eggs. That would have made some headlines. But I had tenure. I was in no hurry. After that, I did what I call the pool party experiments. So what we did was, because only 10% of the genetic males turned into females, so we asked, what about the other 90%? Are they, can they reproduce successfully? And so we did what I call these pool party experiments. And this is, I see Andy Blausine is in the audience. This is some Bruce Waldman training right here, right? So we did these experiments where we put 4 females in one of these tubs, 4 control males, 4 atrazine treated males. And we just asked, can these atrazine treated males compete with the controls? And so it's a really simple assay. We put them in there at 7 p.m., so there they go. They're happy. The lights go out, put on a little Marvin Gaye. Young people don't know nothing about that, but set the mood. And then we had stitches in everybody so we could tell who was who. And then the end of our assays, we just simply came into the morning and asked who got the hookup, who got the female and who didn't. We did this 4 times. Actually, we did it 5 times. But one morning, a student kicked the pool, so we didn't get data. She had to find another job after that. But I'm only joking. But we did, in 4 trials with different sets of animals each time, we found that the atrazine treated males almost never get the female, almost never. So then I'm an endocrinologist, not a behavioral ecologist. So that was my Harvard training, the behavioral ecology part. My graduate training told me, well, you got to measure something. You got to stick a needle in the heart and measure something. So if you measure testosterone, the controls on average are much higher than the atrazine treated ones, as you might guess. If you look at the individual data who make the love connection, you'll see that these poor atrazine treated males, it's almost like there's a threshold. It's almost like if you're below this level of testosterone, either the females don't like you or the other males beat you up. But either way, you're not successful. Okay, so they're the two hearts. They're the two successful atrazine treated males. So these atrazine treated animals just don't have enough testosterone to be competitive in the pool. So I probably could have published that, but then we did another series of experiments that I called the Motel 6 experiments. In this case, I just got them a room and I asked, okay, here's a male and a female, here's an atrazine male and a female, etc. We let them have, a night, and then we collected the eggs, and then we just literally, well, I like to tell you there was some fancy technique involved, but literally there was a student sitting in lab going 1, 2, 3. I think he became an accountant. Actually, I'm being obnoxious. He's actually an assistant district attorney now. So turned him away from science, I guess, or at least this kind of science. And the idea was we counted how many eggs developed, as you can see in these panels, versus how many eggs were infertile as a measure of fertility of these males. And we found out that while controls fertilize about 85% of a female's eggs, atrazine-treated animals only fertilize about 15%. So they can't get the females in the pool party. And even if they do, even if you get them a room, they can't, they have very low fertility because one, they don't even try. In fact, we have another paper coming out now from a graduate student showing that those atrazine exposed males with the high estrogen, they prefer to mate with other males. So they aren't even interested in the females. And even in the Motel 6 experiments, even if they try, here's what the testes look like under the microscope of a control and atrazine treated animals. And you can see we did fancy statistics and measure, but you can see the difference because what you're looking at in control, see all this in the middle? That's the testicular tubule that I'm outlining. And those are all mature sperm in those tubules, whereas these atrazine treated animals have testicular tubules that are full of cellular debris. You don't see any of those dark sperm heads. And so we have quantitative data, but you can see that they're different. And then we published another paper. In this case, atrazine induces complete feminization and chemical castration in male African clawed frogs. The company hates the term chemical castration. That's why I put it in the title. That's the kind of brother I am. The other thing that's important is 9 undergraduates, mostly people of color, 9 undergraduates co-authored this paper, and every one of them now has either a PhD or an MD or both. And I'm equally proud of that. So here's what we're doing now. I'm only going to tell you briefly. What we're doing now is we've taken different lines of African clawed frogs. Okay, so this is work that we're about to submit now. And so what you're looking at is each one of these bars is a family of frogs, is a sip ship produced by a different pair of frogs. And they're all genetically male. And the blue show you the ones, and these were all exposed to estrogen. Okay, so this whole graph, they're all exposed to estrogen. They're all genetic males. There's about 100 animals in each bar. And the blue are the genetic males that develop testes. And the red are the genetic males that develop ovaries that were sex reversed. So what we've shown is that depending on which family of frogs you use, and this is highly repeatable, we've done this with the same pair of frogs over and over again, you can get any answer you want. I can take this family of frogs on the left and treat them with estrogen and show you that estrogen is not estrogenic because less than 5% of the frogs are sex reversed and develop ovaries. And in the same room with the same students, I can give that same dose of estrogen to this family of frogs and 100% of them will be sex reversed. So there's a 10,000-fold difference in sensitivity between one family of frogs. And by family, I mean set of parents. There's incredible variability. But what's more is all of these on the left that are insensitive are from commercial sources that you order online. All of these on the right that are sensitive, you can separate them statistically, are wild or feral animals that either I collected in California and San Diego, or animals that I collected in South Africa. So the animals that you buy that are supplied to universities are incredibly, relatively insensitive to animals that you collect from the wild. And we're excited about this for several reasons. One is we're interested in the variability. Whereas most people who are doing toxicology or cancer research, they want repeatability. They want the same answer every single time. That's why they use this strain of Sprague-Dawley rat. But if you're studying Sprague-Dawley rats, you're studying Sprague-Dawley rats. You're not studying rats. The things that you learn about Sprague-Dawley rats don't tell you very much at all about rats at all. If you're studying breast cancer and you're studying MCSF7, so first and foremost, the animals that we adapt to the laboratory, we adapt because they're adaptable to the laboratory. So you're not adapting, you're not using the typical animals anyway. And the same goes for cell lines. The cell lines that you're using because they become immortalized. You're using them because they're unusual. And you're using them because you get the same answer every time. But even if you find a cure for cancer from that one cell line, not everybody's breast cancer is the same. And I'll talk about this later, especially when we start to talk about minority health. With the exception of HeLa, none of the cancer cell lines that we use come from Black or Latinx individuals who are more likely to get and die from most of the cancers that we're concerned about. Now, back to the little boy who likes frogs and the frog problem. If this line of frogs on the left, which are all laboratory, same species, right? Just different families, can't even tell you something about this family of frogs on the right, how can we use Xenopus, the lab rat of the amphibian world, to make predictions about how atrazine and other chemicals would affect other species out in the wild? So we set off to try to address this. Actually, we didn't have the Xenopus data at the time. Those are brand new data, unpublished data that I just showed you. But we did look at the time at leopard frogs, and this is from work I published in Nature. So here's testis, and these don't exactly become hermaphrodites, but they grow eggs in their testis. And you see all this, I usually call it the junk in the trunk. This is a male frog that's actually yolked up eggs that are bursting through the surface of its testis. And that was produced in the laboratory. But we used these North American leopard frogs because we wanted to find out if this was a laboratory artifact restricted to just one species, the Xenopus, or if we could see these type of effects in the wild. So to give you an idea, I haven't talked about doses yet. We're using 0.1 parts per billion atrazine in the lab. This is a log scale I'm showing you. The package of atrazine recommends 2.9 to 29 million parts per billion. So farmers are applying it at levels that are 290 million times higher than we're using in my lab. You're looking now at minimum and maximum atrazine levels in agriculture runoff, temporary pools, permanent water, and precipitation. Here's what we're using in my lab. So all of these environments would be at risk. So we asked this question before we went in the field. We asked, is 0.1 parts per billion out there? In fact, rainwater, as you can see, precipitation, can have 100 times more atrazine in it than we're using in my lab. And your drinking water is allowed to have 30 times more atrazine than we're using in my lab. So we went out to the field, we collected some water at multiple sites, we collected frogs, and here's what we found. Here's kidneys, and these are gonads, and now I'm showing the histology. So just imagine I'm slicing a salami and folding out a slice. The color is different because of the stain we use under the microscope. Now, if I blow that up, and in the Nature paper, we call these testicular oocytes. And the company literally tried to have the paper retracted because they said we made up a word, testicular oocytes. They didn't like the word. My response to the editor was, aren't all words made up before they're actually words? Plus, I went to Harvard. I can make up a word. So here's the idea. Here's what we published. You see all the red areas. I'm not going to call them red states, but the red areas here are areas where most of the atrazine is used. The gray-white is, there's a scale. And so we did a study, and I usually joke that we control for latitude, but the reality is this is Highway I-80, and we were going to a conference in Indiana to a HERC conference, and we collected frogs and water, and we showed that everywhere we found atrazine, we found hermaphrodites and vice versa. But we also showed that we could take animals from a red area and raise them in the lab, and they wouldn't develop as hermaphrodites. And we could take animals from a blue area and expose them to atrazine, and they would develop hermaphrodites. So we didn't just show correlation, we showed cause and effect. This is a more recent map of atrazine use. And now we've done this huge study And somebody once asked me, how come we're not studying the South? And that's only because leopard frogs, the range for round Amphibians is only this area. And this area here where my arrow is, they're very scarce and it's hard to get permits to collect, especially in Illinois. So we didn't want to collect in places where frogs are already in decline. At any rate, that study is ongoing. We're also doing some very narrowly focused studies, and I'll stop after this and ask if there are questions. Um, Almost like a dream come true, I found out Congaree National Park is the only national park in South Carolina. And it's the place that I grew up. It's the place that I fell in love with frogs. I mean, this is where I became the little boy who likes frogs. And a few years ago, this article came out: chemicals and medicines tainting Congaree National Park waters. And it was almost like, I used to make this joke that I took the hood to Harvard. Now I can bring Harvard back to the hood, because it was almost like, wow, I get to come home. I get to come home to the place and do something positive for the place I grew up in. Because this whole article was about, you see the photograph? The whole article was about, oh, by the way, the chemicals that are tainting the park are, you guessed it, atrazine and ethinyl estradiol, atrazine from agriculture and ethinyl estradiol from the leaky septic tanks from the low-income Black community that lives around in the swamp area. And the whole news article is about, what about the white person who comes to kayak through on the weekend? What if they fall in the water? The whole article was about that. And my response was, what about the almost 30 species of frogs that are living in the park, the reason the park is there in the first place? And what about the low-income Black community that's living around the park and exposed every single day? So I got funding to now study the frogs in Congaree National Park, and to kind of dip over into public health. Before I do that, are there burning questions that I should answer, or should I go more into the human and canary aspects of the talk?
So Tyrone, I can ask the questions for you, but if you look at the bottom of your screen, there's a Q&A button that you can press.
Okay.
If that doesn't work, I can read them to you.
Okay, so are estrogen molecules absorbed through the skin? And frogs, absolutely, yes, they go through the skin. And in humans, they do too. You might know that there's several different types of birth control. For women, that's a patch that goes onto your back or lower hip or wherever you want it, and you absorb those compounds through your skin, and then they become systemic and regulate your reproductive physiology. So another question is, do the frog patterns vary? They do. Each individual, it's like a fingerprint. And one of the things I was really interested in until I got involved in atrazine was trying to study sort of cell fate, and how this cell knows it's part of the spot and this cell knows it's part of the background. But each frog's pattern is unique. In fact, I used to take pictures and identify them by their patterns. Says company really want you to do the science. When you did the science because the science made them look bad. I'll talk about that, about the company's response. Do you have any idea what the mechanism is for why those few atrazine treated males were not demasked? So this is an interesting question. So somebody's asking, do I know why some frogs respond and why some don't? Even better, and when you look at that chart I showed you with the estrogen exposed frogs, where some families don't respond at all. I don't know the answer to that, but there are 2 people in my lab right now whose PhDs are depending on finding the answer. So some of it might be how to take up and metabolize the steroid. We don't think that's it. We think that there's a mutation in the male determining gene that makes them more susceptible to the estradiol. And the reason we think that, rather than something with the estrogen receptor, is that we've inbred that really sensitive line for now 6 generations. And in the 4th generation, we start to get spontaneously sex reversing animals. So we now have lines of frogs that have no female chromosome, no female determining gene. You don't have to give them estrogen. They're genetically male, and 100% of them will develop ovaries. So we've actually evolved a new sex determining system in the laboratory. And so the exact molecular mechanisms, we're looking at gene expression, et cetera, to address that question now. But the question is probably relevant to why some people get breast cancer and some people don't, why some people can be exposed to the same chemical and some people get sick and some don't. And those are broader questions we're interested in. Generational changes in the frogs, yes. Like I said, we can select for sensitivity. So we can crossbreed the resistant ones and the sensitive ones and select for frogs that are more sensitive. We have some super males that actually can reverse the sensitivity, and we think it's related to a gene called DMRT1. Talked briefly about similarities between chemical causing genes in frogs and breast cancer. Are CYP enzymes similar to both frogs? So yes, I'm going to talk about that in just a second. So the question is, are the CYP enzymes in frogs and humans similar? And I'm exactly about to talk about that. So this is a photograph that I took in a place called Lake Nabogabo in Uganda, and I took it because I think it clearly shows what I call the oneness between environmental health and public health. The runoff from this crop, which I can't identify, I think it's arrowroot, goes into this containers, and that's the drinking and cooking water for that village. And I like this because it shows how, if there was something in the water that were affecting the frogs and the fish, the people who are drinking that water straight out of this pond next to the lake would clearly see the connection between human health and environmental health. Whereas in the way that we live, so here's my village, Oakland, and I live somewhere there, but my water just comes from there. We make all these assumptions about government regulation and that there's nothing bad in our water. In fact, all you have to do is ask people in Flint, Michigan to know that that's not the case. So sometimes I've called this from silent spring to silent night, because I think in much the same way that Silent Spring taught that the pesticides that were causing the death of birds and our Silent Spring was a warning to us, I think in a very similar way, our pending Silent Night, or the death of frogs, for those of you who don't know, up to 80% of all frogs are in decline or threatened. A group of animals that outlived the mass extinction that took out the dinosaurs are now facing global declines. And in much the same way, I think our Silent Night is a warning to us. I don't think, and I would never say that pesticides are the reasons that amphibians are declining, because we've done a lot more than put chemicals into the environment. So this is a paper I published with a couple of graduate students a few years back, showing all what I call level 3, the ultimate reasons for amphibian decline, which include introduced pathogens, environmental pollutants, atmospheric change, climate change, habitat modification, invasive species. And it's showing the interactions between these two and how these show up on the next level. Even though chemicals can have a big impact on amphibians, there's no single cause, I would argue, for amphibian declines. But when you look at a situation where there's habitat loss, I don't think anybody would argue with me that habitat loss has the biggest impact on amphibians and wildlife. But if you're a frog and you've lost habitat, let's say in California, and the only water available is agricultural runoff, as is the case in many places in California, or chemical contaminated runoff. And when you're talking about chemicals that lower immune function and negatively impact reproduction, while chemicals may not be the cause of global amphibian decline, they're certainly in the middle and involved with many of the other causes. I published a paper a few years back with several colleagues to address the following. Glenn Fox once said, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. So I've shown you, because I got in these arguments with Syngenta, I've shown you that not just correlation, and I've shown you data from more than one species, genera, even phylogenetic family of frogs, but similar data have been produced in every vertebrate class that's been examined. And so somebody was asking me about the P450 enzymes. If you're not interested, just ignore this slide. But this slide is showing how aromatase is normally regulated. So aromatase is a cytochrome enzyme, cytochrome P450, CYP19, and it's regulated by CREB, which is cyclic AMP dependent. So what atrazine does is it binds to the phosphodiesterase that allows an accumulation of cyclic AMP that allows activation of CREB and overexpression or inappropriate expression of CYP19 aromatase. Now, other labs have shown this. I was involved in co-authored 2 papers with a group from Japan that also supported this molecular model as well. But it's pretty well established that that's how atrazine works. That atrazine down there is an indication that we also suggested that atrazine binds another transcription factor called SF1. And it turns out that's probably not true. That was probably an artifact of some of the molecular biology. But the interaction with phosphodiesterase is important. So I published a paper with 32 scientists from 12 different countries. And we got together and we put together our data to show that this wasn't just a problem that only occurred in frogs. So for example, here's my frog testes, give it atrazine, no sperm. A group in Belgium showed the same thing in fish. A group in Argentina showed the same thing in caiman. It's a reptile like a big alligator. Groups in Nigeria and in Croatia, I didn't know who any of these people were, showed that if you look at a rat's testis, so there's one full of sperm, give it atrazine, no sperm. And this is a research in Pakistan that showed that sperm in the testes of a bird, give it atrazine, no sperm. So this effect occurs across vertebrate animals, across vertebrate classes, and experiments that are all done in very different ways. We asked whether or not the reduction in sperm was because testosterone was reduced, and you need testosterone to develop sperm. This is a group in England who examined salmon, fish. This is my work in frogs. This is work in rats. So atrazine reduces testosterone across vertebrate classes in every study that's been conducted. Now, if you look in humans where you can't do experiments, what my colleague Shana Swan showed in Columbia, Missouri, that there's a significant correlation between atrazine levels in your urine and low sperm count and inability to get your wife pregnant. And just as a matter of fact, you'll notice that 0.1 parts per billion, the amount that it takes to chemically castrate a frog, if you have that in your urine, then you have a low fertility and a low sperm count. Now I'll smash the data down. So I would argue that frogs are making these predictions and they're bearing out. So I've smashed the data down because a study in California in 1993 showed that these are field workers. These are people who work in the field. These are their atrazine levels. Okay, I'm going to smash it down again because these are atrazine levels of men who apply the atrazine in California. So men who apply the atrazine have 24,000 times the atrazine in their urine than we know is associated with low sperm count in men in Columbia, Missouri. They have 24,000 times the atrazine in their urine that we use in my lab. So one of these guys could pee in a bucket. I could dilute their urine 24,000 times, and I could use the atrazine in their pee to chemically castrate and feminize 24,000 buckets of 30 tadpoles. We know nothing about the reproductive health, and for a great deal, we don't know much about their health at all because they're primarily Latinx workers. Many of our agricultural workers have life expectancies of 40. And in addition to atrazine, they're exposed to chemicals like chlorpicrin, which was originally developed as a nerve gas in World War II. They're exposed to chemicals like 2,4-D, which was a component of Agent Orange used to destroy food in Southeast Asia. So I'm just a little boy who likes frogs, but I've gotten very interested now in environmental justice and environmental racism and how many of these environmental influences may help explain the health disparities. For example, this is California. You're right next door, so you probably know these facts. We're well, I don't know where we stand now, but the last time I checked, we were the 5th largest economy in the world. Not because of tech and not because of Hollywood, but because of agriculture. 1 in 10 jobs, 30% of the land, 350 agricultural products. Half of the US's food comes from California. I didn't know this. 50% of the entire country's food comes from California. We use more pesticides than any other state, and 90% of their workers are Latinx. So if I put on here now the top 10 counties for agriculture, that's about 30% of the land. Where do you think the 30 poorest towns are in California? So the people who make us the 5th largest economy in the world are the targets of chemicals that we know are associated with adverse health outcomes. Again, I'm just a little boy who likes frogs, but it makes me see my work in a very different light. Does atrazine turn on aromatase like we showed? In our frogs, it causes vitellogenesis and oogenesis. So egg yolking and egg production. That's probably not going to happen in mammals. In mammals, what you're worried about are mammary tumors and prostate cancer, which are both aromatase and estrogen dependent. With regards to prostate cancer, there's an 8.4-fold increase in prostate cancer incidence in their factory in San Gabriel, Louisiana, a community that's 80% Black. I'll tell you why I bring that up in a second. There's at least one study that shows a very strong correlation between atrazine contamination of well water and women's likelihood of developing breast cancer. And that's just correlation. But if you look at rats, again, testosterone is decreased with atrazine exposure, estrogen increases. And this is work that actually the company did themselves. They showed that if you give atrazine to rats, there's a concomitant increase in estrogen-dependent breast cancers, breast tumors, mammary tumors in the exposed rats. What's more, in humans, if you take human cell lines, and we published this, but another group has published it as well. In fact, 2 other groups. If you take a human cancer cell that doesn't normally express aromatase or make estrogen, so it's normalized to 1, if you give it atrazine, it expresses aromatase and starts making estrogen. Just like we've shown in fish, amphibians, reptiles, birds, and rats. Human cells have the same, as somebody asked, CYP19, cytochrome P450 19 hydroxylase, regulated the same way as fish do. So I went to see them, and then I'll stop in a second for question. I still think they should switch the Y out with an I in their name, but anyway, that's just me. They have a pipe that flows directly into the Mississippi River. 1.2 million pounds of atrazine flow into the Gulf of Mexico every year. And a community that looks much like this, and as I said, it's 80% African American. And Syngenta is not the only factory that's down there. Here's why I bring this up. If you look at the top 13 cancers that you're likely to get in the United States, you're more likely to get 11 out of 13 if you're Black. And this is corrected for access to healthcare, socioeconomic status, and I can show you similar data for Latinx individuals. What's more is if you look at mortality relative to white or Caucasian Americans, you're more likely to die from 13 out of 13 cancers. Now, my colleagues who study cancer, who are experts, tell me that less than 30% of cancer is explained by genetics. So when your doctor tells you you're more likely to get breast cancer if your mother, your sister, your daughter, somebody in your family has it, they're not telling you that you have bad genes. And there are some genes associated with breast cancer, but like I said, less than 30% is explained by those genes. They're not telling you you have bad genes. They're telling you that you've been exposed to the same crap as the rest of your family. And from what we're seeing in our frogs and what other people are seeing in fish and rats, even if you move away from that contamination, from that exposure, there are epigenetic, there are transgenerational effects that will probably move with you. One of the other things I discovered, this is, we don't have to discuss this too much, but this is a reverse transcriptase PCR showing that breast cancer cells, when given atrazine, express aromatase just like everything else we've studied. This was work from a graduate student. That's significant because aromatase, as I told you, is really important in breast cancer. You produce estrogens locally that bind to the estrogen receptor and cause those breast cancer cells to grow. That aromatase is so important that the #1 treatment for breast cancer right now is a chemical that knocks out aromatase so that your damaged cells don't see estrogen and grow. How crazy is it that the #1 contaminant of drinking water and groundwater does exactly the opposite, promotes aromatase and promotes breast cancer? In fact, the same company, Novartis, up until 2000, made both chemicals so that if you were living in the Midwest taking exposed to atrazine that's associated with breast cancer, it was fighting it out with a pharmaceutical made by the same company that does exactly the opposite. If you were taking letrozole to treat your breast cancer, it was fighting it out with a chemical that does the exact opposite, made by the same company. So I think that as a little boy who likes frogs, my interest in this aquatic organism has taught me quite a lot about this aquatic organism. Even though I didn't set out to think about humans and human health and environmental justice and environmental racism, I think that's where I've ended up. I want to tell you one last quick thing, and then I'll stop for more questions. The one thing that has impacted me the most, more so than my work, is studies in rats. And just so you know, I'm pretty sure I study frogs because I'm a little boy who likes frogs. I'm pretty sure people who study rats aren't little boys and girls who like rats. I'm pretty sure they're trying to learn things about humans. And what struck me the most, even more than my frog work, is that studies have been published showing that atrazine causes prostate and mammary cancer in rats. It causes immune failure in rats. We've shown the same thing in frogs. It causes neural damage in pregnant rats. And the study that impacted me the most, studies were done by the EPA, peer-reviewed and published. They showed that rats who were exposed to atrazine while pregnant were more likely to have abortions, miscarriages. They showed that of those rats that don't abort, the sons are born with prostate disease. They're born with the prostate of an old man. And if those rats that don't abort, the daughters are born with impaired mammary development, such that when they grow up, and this is what their mammary glands look like, when they grow up, their offspring have retarded growth and development because their mothers can't feed them properly. So think about that. The rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. And again, we're seeing similar transgenerational type effects across frogs. And so when I think about my son and my little girl, she's now 25 and thinking about having kids, when I think about the idea that my grandchildren could be affected by chemicals that we're using today, your grandchildren could be affected by chemicals that we're using today, it makes me realize that I have a much bigger responsibility than just a little boy who likes frogs. So I'll stop there given the time. There are questions, and I guess I can answer questions now or wait for the meeting tomorrow, whatever your preference.
You should answer questions now, Tyrone, because the meeting tomorrow is only with grad students.
Oh, okay, okay. Okay, so the CYP enzyme one, I already asked. I'm a people person too. I'm used to seeing people interacting. So it says, I noticed the atrazine levels are pretty high in the Portland Corvallis area. Do you know if this thing has been causing problems? There's a group in Oregon actually who's been working very hard to gather published data and working for legislation to ban atrazine. So there are problems that are noted. And most of the atrazine use in Northern California and Oregon, the green that you saw, that's not corn. If you know anything about that area, that's all used in forestry. So it's a very different type of use where it's not applied every year. When they clear cut, they use a lot of atrazine and then they might not use it again for another 20 years in that one particular spot. So it's a very different pattern use. But there are some birth defects and things that I didn't have time to talk about that are associated with atrazine that are being litigated right now. Did the changing sex between the African clawed frogs also correspond to the development of different musculature throughout the frogs as well, or were the changes only found in the sexual organs in the voice box? So there is another muscle, and pardon me, I'm not an anatomist. Marvelly Wake would kill me. There is another muscle in the frog's sort of chin, if you speak, that's sexually dimorphic that we published on in another article. But other than that, I don't know of any other musculature changes. We did report in the second PNAS paper on not just the larynx and the gonads, but we reported that some frogs maintain oviducts, even though they're genetically male, when they're exposed to atrazine. And we also measured breeding glands. So you can have male frogs, the testosterone-dependent breeding glands don't develop properly. And I had a student trying to examine the brains and associate that with the behaviors, but she's been unable to identify she can identify sexually dimorphic areas of the brain, but she doesn't find that the brains of atrazine-exposed males are more similar to females. So in regards to atrazine in urine, is it atrazine that is excreted or it's metabolites? So the atrazine that they measure in the urine is, it is a metabolite. It's called, I think it's a glutathione addict. And actually for that reason, it's been suggested that those estimates are an underestimate because there are other metabolites in the urine. So they typically only measure one key metabolite. And it's been suggested that that's an underestimate because they're not measuring the total metabolites in the urine. Oh, hi, Becky. What is the status of banning atrazine in the US like in Europe? So atrazine is now, as of about a month ago, banned in Hawaii, Alaska, and Puerto Rico. I think it's also regulated in Wisconsin, but there is not a federal ban on atrazine. We'll see what the new administration brings. But the EPA has issued a report. It's about that thick, where they acknowledge that atrazine is a reproductive toxin and is a danger to wildlife and humans. And in California, under Prop 65, it now is potentially restricted, and it has to go out with a warning notice from EPA. Do you think decisions on atrazine are explained by lab strains of frogs that don't respond the same as wild frogs? I think I'm really excited about. Maybe nobody else has this, but that red and blue graph that I showed you, I feel like if that's the last graph I show before I die, I'll be satisfied. And just looking at that, and looking at the fact that Michael Gordon, looking at the fact that the company and everybody else is using these animals from from these companies, and yes, you get a very different answer. We've shown it over and over. I've only shown you that one graph, but we've shown that it's repeatable. So we can take the same pair and breed. In fact, you can come to me and say, Tyrone, I treated with this level of estradiol, with this concentration, and I got this sex ratio. I can tell you who the parents were. Whereas my students who are doing the molecular biology, they can come and say, okay, we treated with estrogen and we got this much vitiligin and gene expression. There's so much variability. But if you tell me the dose of estrogen you used, and you tell me the sex ratio, you tell me the outcome, I can, within one or two pairs, tell you exactly which parents you used for that experiment. That's how repeatable and how robust those data are. So yes, I think, and it says that we can't use that model lab species to tell us anything about what's happening in the wild, let alone, keep in mind, like in Congaree Swamp, not only are you exposed to atrazine, you have a combination of ethinyl estradiol, so you're exposed to more than one thing. Some species breed in the winter and some species breed in the summer. So some species are being exposed to wintertime levels, but also to lower temperatures, which will decrease metabolism and uptake, et cetera. Other species are exposed in the summer, they develop faster, they're exposed to different levels, they're also exposed at a different temperature. So Xenopus laevis doesn't allow you to predict any of that kind of stuff that would allow you to predict what's happening to amphibian populations in the wild. Sorry, I feel like I answered those questions really fast. So if anybody has other questions or needs clarification, please feel free.
So are there any other questions for Tyrone before we end the webinar?
Thank you. Thank you.
So where can we find the recording? I will ask Teresa about that. But Tyrone, thank you so much for—
It was my pleasure. Thank you.
It was a pleasure. I know you're meeting with the grad students tomorrow at 10.
At 10.
Okay. And we look forward to continuing to interact with you, hopefully in the future.
Thank you. And I wish I could have come in person.
Yes. When this is all over, we'll have you over again.
I'd absolutely love to.
Okay. Thank you very much. Goodbye, everyone.